to kind of go through a little bit of what CRISPR is and how I've told my students and how I break. Yeah, so this is the PG Ed's CRISPR lesson. So I'm going to skip some of the scenarios, but I mean, I'll read them to you because this is what I would do if there's like a whole class presentation. So we ask students to think about this question. We say, well, you've been offered a deal from the genomics company. You can get a free genome sequence and analysis of all your DNA that includes a report of your ancestry, traits, and medical profile. The medical profile tells you about diseases for which you have a low risk of getting, and also those you have a high risk of getting. Are you interested in this technology? Why or why not? So we kind of have students talk about that for a little bit, and then we follow it up with a second question or a scenario, which is, for the first 100 volunteers, the company is offering to correct several of the disease-related genes found by the analysis. Imagine this were a very new procedure approved by the government for safety, but without a great deal of long-term study. Would you volunteer for this service? And we have students kind of talk about that, and we do 4 corners and talk about the ethics, pros and cons, and what they're thinking. So it's kind of just to get ideas out there. So this is when we start introducing this idea of what gene editing technology could be used for and how we might see it posed in the media, or when we start to see it advertised, how we might start to see it advertised. And this is kind of things you may start to see if policies kind of develop and laws pass. So this was kind of, getting students thinking about it. And then I like to give them a little background about CRISPR, which a lot of people don't realize, but CRISPR is old. It's not new. It's actually pre-existing bacterial immunity. It's what bacteria use to prevent infection from viruses. So here you can see this is a phage virus, and this green stuff is like genetic content. So this phage virus is adhering and infecting the cell, or injecting this content. And what you have here these are all CRISPR. So you can see here you have this spacer DNA, and you have these CRISPR sequences. So the spacer DNA is derived from. So you can see over here. So you have this clustered. So CRISPR stands for clustered, regularly interspaced, short palindromic repeats. And here are the repeats right here. These little black sequences, and the repeats are just known sequences of DNA. They're palindromic because they read the same forwards and backwards. So like if it's ATCGA on this side, it'd be ATCGA on the bottom side because DNA is complementary. So the base pair, so they have to read, the reading frame to the right has to be the same as the reading frame to the left in order for them to match up because they're complementary. So here's that kind of repeated over here, these clustered regulatory repeats, these little black kind of diamond. So what these are the CRISPR sequences. These different colors represent past infections. So this cell, this round cell, is the bacteria. So this orange, this purple, this, I guess this is pink, this purple, this orange, and this blue, these are past infections that this bacteria has incorporated and has a memory for. So kind of like memory B cells, where you've seen a pathogen or infection, you have memory cells for it, where your body remembers it, and if it sees it again, it can fight it. This works in some bacteria have a similar mechanism, where they were able to remember some of these past infections, and they incorporate it into this kind of rudimentary immune system right here. So here's this green virus, and here's going to be this incorporation of it as it defeats it. And this is going to be a memory. So what this creates over here, this green, this RNA is going to get processed, and here's an RNA transcript as the cell processes it. And what happens here, you have these molecular scissors, kind of like restriction enzymes, but different, that will bind to and cut this sequence out and rendering it harmless. But now, if this bacteria sees it again, it'll survive this because it has defeated it once, and it now has a memory for it. If it didn't defeat it, it would have infected the cell, and this would create more host viruses. But it is able to defeat it and cut it out. And here you can see these little scissors, and what they're doing is they're cutting out, targeting, destroying this green segment only right here. So this is already in existing. This is actually natural CRISPR in a sense. This is what bacteria have done, but humans, Jennifer Doudna, actually she's the reason why I'm here. She gave a grant to PGED to hire me to do some community outreach to bring PGED outside the classroom. So rather than just going to schools and students, taking these ideas into the community community workshops, parents, families, not just in school, because some people haven't been in school for a long time. They should know about this technology and how it can be used. So that's kind of my role here at PGI this year. But so people, like Jennifer Downer and a lot of others actually realize, wait a minute, this is cool, that this ability to recognize a very specific sequence, target it, and cut it out. And they said, wouldn't that be cool if we could do that, cut it out, and also insert a desired sequence? So the bacteria are able to kind of recognize and cut it out. What if we can take this a step further and cut it out and insert a different desired gene sequence, which is what synthetic CRISPR technology does. Mm-hmm. Over here, this whole thing is DNA. Rather than showing individual bases, they just did as a squiggle. Here's DNA. Here's this red POW symbol. So this is the base to be changed, or the segment that, for whatever reason, you don't want. Maybe it's dangerous, or maybe it's just not desirable, but you want that cut out and corrected, a different sequence put in. But if it's for a disease, or maybe if something as cosmetic as eye color, whatever it is, you want undesired sequence taken out, and you want a desired sequence put in its stead, using this kind of this. So here is the nuclease, which is the Cas9 nuclease, which is just a protein. It's an enzyme, and it has a guide RNA. The reason why it has a guide RNA is just so it can base pair. Mm-hmm. So you need that base pair. You need to have because it's all about molecular fit and size. If you have an A, you need to base pair to a T. If you have a C, you have to base. So the RNA does it, and it's written in RNA versus DNA, so that when it gets transcribed, it can create a protein.
So the DNA is double helix, so it's like bound together. So how does the guide RNA identify?
So what this is kind of simplifying and oversimplifying is this would be rather than saying a whole DNA sequence, this would be really an RNA transcript. It would be a segment. They're showing you the whole DNA sequence, but really in real life, it would be a transcript, an RNA transcript that would make that protein. Or you could also do it too, where you have the guide RNA, but you can give it DNA bases to base pair to DNA. So there's two kind of mechanisms, but this is kind of oversimplifying it, just to show you that they use a guide RNA to base pair, but they're not showing the exact step-by-step processes, only because it gets a little bit more complex. Mm-hmm. So what happens over here is this binds to it using the Cas restriction enzymes, but using this kind of the scissors, these molecular scissors that work sort of like a restriction enzyme. They're able to cut it at the exact location, able to bring in a desired sequence. So see how they changed now DNA instead of RNA, just to show you that it's this genome, but it will be transcribed into RNA later.
So the sequence you're bringing in is not necessarily attached to the Cas9, it's just something separate.
It is, exactly. But what they're also not showing you here is that it'll be brought in with an enzyme.
Okay.
It's not just going to magically float, into the cytoplasm, but with the help of an enzyme, it will be inserted and added to this sequence here. So now when this gets transcribed into a transcript, the new desired sequence is there. Would be there. So this is kind of like a simplified, really, really simple version of what's happening, of course. So this was kind of like the pared down version of like step 1, 2, and 3, but of course it's a lot more detailed steps than that. But I feel like this helps students get a kind of a starting sense of what's happening. What is CRISPR? Again, clustered regularly interspaced short palindromic repeats, and we kind of talked about what that meant, that little short repeat segments are. And so it targets specific section of DNA, which you saw that happen in the little video, makes a precise cut or break at the target site, and it can do one of two things. It can make a gene non-functional, so turn a gene off, if you learn about gene regulation, or it can replace one version of a gene with another. So take out a faulty gene or just an undesirable one to put in one that you do want, that's desired. So that's kind of what the benefits are and how it can be used today. So what are the potential applications of CRISPR to human health? So those kind of two scenarios are one way students kind of get talking about that. But then with this, we go into talking about gene therapy. So looking at sickle cell, looking at being able to, what causes red blood cells to be sickle shaped is just one nucleotide, just one. So imagine being able to cut that nucleotide out, put in the correct sequence and over time make less and less and less of that sickled cell. So it wouldn't be gone overnight, but your body would make less of it as you correct some of those cells, and those newly corrected cells replicate through mitosis.
So you would insert the CRISPR into single blood cells, red
blood
cells?
A few, like a series of a few.
Yeah.
So when those blood cells reproduce, you have more. So yes, using as gene therapy to over time reduce sickle cell, and then within that patient's life, they would eventually produce no more. Or cystic fibrosis. It's an overproduction of mucus in the lung. So you have an enzyme that overproduces fluid or mucus. And so this enzyme, this protein, constantly makes mucus, which is basically like having pneumonia, where it's like you're drowning, So being able to edit it, again, same kind of tech, same kind of system, being able to edit out and to make that gene non-functional. If you can make that enzyme make the DNA not make that enzyme, so it'll never be transcribed, never be translated into that enzyme protein, a form of gene therapy. So you can make it non-functional.
So if you want to make it non-functional, you can just make the cut. You don't have to insert other.
No, so you can. So exactly, you can just edit it out, take it out. So that's the difference. But all that is really just one base pair difference. Having an A, for example, instead of a G, all of a sudden having A means now you make mucus. Having a G means you don't. So let's take out that. So exactly, taking out that A or switching the A to a G. Another thing that's being done is, gene editing to cure type of liver disease in adult mice. So again, curing diseases. This is again only done in mice. This hasn't gone to clinical trials because it is dangerous, and no one wants to be the guinea pig. Very few people, you don't know what the outcome is. It could be therapy or it could go horrifically wrong. And it's unethical to really say, we're going to test on you, but, you might not make it. You can't do that. So which is why it hasn't gone to human test trials, just because it's still new and it's still being developed, and there's still so much room for error right now. But in mice, it has been, it is able to cure, liver disease in mice. Another thing that CRISPR can do is help with the shortage of organ transplants. So right now, pig organs are relatively very similar in size to human organs. So size and their mass and their weight, and generally speaking, they're very, very close to us. CRISPR is being used to kind of edit out some of these, what they call PERVs or porcine, viruses. So I forget what the full acronym is for, but it's like porcine. The E is for endoretroviruses. And so they abbreviate it PERVs, which is kind of a funny name. But CRISPR is able to actually kind of edit out those viruses so that you can make these organs usable for humans and risk-free of having these viruses that are associated with, because if you're using a pig organ, pig diseases now come with it. So, but if you can make sure you edit out the risk for those diseases, you can make the organ actually a little bit more adaptable for a human body, which is what is being done now. One of the ethical concerns is that can everyone use a pig organ? For example, people of the Jewish faith, and of the Islamic faith don't consume pork, but would they be able to use a pig organ? It's an ethical concern that PGED actually has talked about, and actually George Church was saying because of that, they're actually looking into goat organs. Goats are a little smaller, so their organs are slightly smaller, but they're still looking at using they actually are considered still a model organism for humans, and they are currently using CRISPR technology and doing research on goat organs to see if they can also be a viable source. But actually, before I keep going, just some of the things that come up with this is that people, one of the questions that comes up with this, what you might think, do you know, so sickle cell, so having 2 copies of this is lethal, shorten your life. Having 1 copy, so this individual we're looking at right now, you can see they have round blood cells, and they have some of these sickled. So this person's a carrier. This person's a carrier. Being a carrier is actually beneficial. It's an adaptation. Everyone's like, oh, sickle cell's bad. Actually, it's not. It's an adaptation. Having the trait is an adaptation. Having 2 copies of it is not beneficial. So the reason why it's beneficial is because we find people with sickle cell anywhere where there's malaria. So, and people always think, oh, that's just Africa. So no, so people with the sickle cell trait are found anywhere where there's malaria. So that's West Africa. That's the Mediterranean as well. So people of Sicily, Greece, the Azores, and Southeast Asia. So India, Pakistan, Thailand, Cambodia, Vietnam are also areas where sickle cell is found, because those are all areas where malaria is. The reason why it's an adaptation is because the mosquitoes, if they, the mosquitoes don't carry malaria, it's the virus that does. So when the mosquito kind of, pokes you with its proboscis, it doesn't transmit anything, it's the virus that it carries. So the mosquito is not the problem, it's the virus that's in the mosquito. And that could cause malaria. But the thing is that virus loves to live on full red round blood cells with high amounts of hemoglobin. So individuals who have nice, healthy red round blood cells are less likely to survive. Individuals who have some of these make it slightly inhospitable for the virus. So being a carrier makes you more likely to survive malaria. So being a carrier in those regions is an adaptation. So people are now saying, wait a minute, if we hurry up and say we're going to get rid of the sickle cell gene, it's beneficial to be a carrier in some regions. Are we now going to change, and this is going to be a new mutation in this environment where now people aren't adapted, which could lead to problems? Are you getting rid of a trait that can be beneficial? So what does that cause? How is that going to affect populations? Are we kind of rapidly or artificially moving evolution along? This, where we start editing genes faster than nature would do it itself. So that raises ethical concerns of tampering with genes and editing them out, for the benefit of health and longevity. But what does that do, the long-term costs in terms of evolution? What are some of the implications of that further along the line? Looking at 100,000 years from now, as we start so-called eradicating certain diseases, and getting rid of things from the gene pool, wiping them out. So what does that cause? So that's another question where CRISPR sounds fine and dandy, but what does that look like 100 years from now? What are the repercussions of all this tampering, And some students have also said, oh, is it going to make us weaker or, less susceptible to survive other viruses when they pop up or, So those are the questions. So again, this idea of should gene editing be used in hopes of reducing malaria, being able to genetically engineer mosquitoes, male mosquitoes. So a research group was able to use CRISPR to make genetic changes in non-viable human embryos. So we talk about embryos for people who are undergoing in vitro fertilization. We have a fertilized egg that's divided, so this is actually an embryo, and being able to use CRISPR to change the genetic makeup of it. Looking at this embryo and going, it has potential to develop into a human, but it's not yet. Are we able to make edits to the genes? And the answer is yes, you are. And if you can do that, to embryos, what happens, if you are able to genetically engineer embryos and then implant them into a woman's body, and she's able to kind of, in a sense, pick some of the genes she wants or doesn't want? Do we stop at diseases? Does it go further? Who decides? How do you decide? And who has access to this technology? Who's going to be able to afford this? Is it going to create a new ism? So all these other kind of ideas and genetic discrimination, or genoism kind of come up of, well, guess who's going to have access to this? Not everybody. The wealthy, and who's not going to have access to it? And who's not going to be able to afford it? And is that fair? And so, again, it raises a lot of questions and concern. It sounds great, but will it be available to all? And this is a cool one about the woolly mammoth and using CRISPR to kind of, in de-extinction. The woolly mammoth, George Church and a couple others in the Yakutia region of Russia have been working on this for years, and they've realized that the woolly mammoths actually helped, helped keep the earth cooler. And so how they did that is because they're so massive, they're about 5 to 7 times the size of an elephant. Their stampede or their stamp or their stamping was able to kind of turn up the ground and the permafrost, re-exposing that ground to cold air, creating a deeper freeze. So every time a mammoth would trample, it turns up the soil, kind of refreezing it. snow, without it, just think of snow. Snow acts as an insulator, keeping the temperature at a constant. But if you can turn that up and expose it to air, it'll refreeze. Turn it up and create a deeper freeze, actually cooling the earth. And so that book, Woolly, that's on the BPS book list, George Church is in there. Ben Mesrick, he's actually a BPS, he's an author, but he's working with some BPS students right now. What they're doing is trying to create a woolly mammoth hybrid. So the Asian elephant is very, very, very similar to the woolly mammoth, except for a few genes. So what they've done is, in the Arctic, there's a lot of fresh preserved specimens and DNA tissue of the woolly mammoth. So they have the full genome of the woolly mammoth. So that what they've done is actually use this gene editing technology to edit the Asian elephant DNA by inserting missing genes that would make it a woolly mammoth. So there's only a few genes that differentiate these two. One, for example, that produces, long fur. Another one that produces, long hair for warmth. Another gene that regulates fat production, so that because woolly mammoth has a lot more fat in a blubber under the skin, the Asian elephant doesn't have as much. So they're able to actually take the Asian elephant DNA, use CRISPR to edit it, and add some of those missing woolly mammoth genes to create kind of a mammophant hybrid. So here's one of the ideas right now is where it's, being genetic technology such as CRISPR is being used to, in rescuing living and reviving extinct animals. So you could see over here, DNA extracted from the mammoth. It's spliced with skin cells of the modern Asian elephant. So here's here, and here's like that missing DNA. So that they did it in dark blue to show you, here's mammoth DNA, here's mammoth DNA, here's mammoth DNA, kind of fused there and spliced in and added using CRISPR. Then the skin cells are programmed to become stem cells or somatic cells. They take and they put that into a blank ova, an ova that has no DNA, except now you incorporate the desired DNA that you want in it. So now because it's an ova and it has a full set of DNA, without sperm, you don't need it, you have a full set of DNA, it will be put into a womb, and essentially an Asian elephant can give birth to a mammoth.
So in this case, instead of taking out a gene, you're inserting
Exactly. More. Well, yeah, exactly. Or you're changing, you're modifying. Yeah. So you'd modify the gene, let's say, for skin, and modify it. So it's not just Asian elephant skin, it's mammoth skin. With that skin meaning it has the ability to grow very, very thick, dense, long fur, and blubber. So it's just modifying, taking that gene that already exists, but modifying it because it'll express a phenotype that they want. And so here it says, embryos grown in artificial womb yet to be developed. So there is artificial womb technology, but it hasn't been as successful yet as they like it. So this isn't happening yet, but you can see it's being worked on, and there's potential for it. So this again, de-extinction, and everyone's like, are we going to bring back the T-Rex and it's going to be like Jurassic Park? I mean, so this is done with specimens that are relatively young, 10,000 years old. That's not that old, We're talking about T-Rex. Those are, millions and millions of years old, and they don't have DNA. They have fossils, but they don't have DNA tissue. You need full DNA tissues. Right now, they just have fragmented pieces that they found. But so there is no bringing back the T-Rex right now, as everyone's, there's no Jurassic Park happening. There is Pleistocene Park, which is where this project is being done in the Yakutsk region of Siberia, Russia. So it's actually called Pleistocene Park. So what's the path forward? Safety, ethics, we talked about that, informed consent, human and ecological health, So even bringing back the woolly, what about the environment that it lives in now? How will that be disrupted? What would the flora and fauna look like there? What parasites will that mammoth have? Have we seen those parasites? Will it affect us? Will it affect the animals there? What are some of the other risks of bringing back an extinct species into an environment that it hasn't been living in for thousands of years? And how will that disrupt the environment? Will it displace other organisms that are living there? Will it bring in other organisms? So there's so many concerns of when you change a habitat with just one thing, all the other things that change because of it. This domino effect, which you can make predictions and hypotheses about, but we can't know for sure. So we say ecological health, that's what we're saying, and human and consent, ethics. Should you be able to edit embryos? Should you not? What will the religious community say about that? Is what happens when you edit it and it's like, oops, it's not looking so well. Do you just toss it out? Like, what do you do? So there's a lot of safety. How safe is it? And start to question, like, what is life? At what point, is it okay to tamper, at an embryo, bigger, more, at a fetus? I mean, it's a lot of questions that this raises. And there was a summit in 2015 kind of bringing this up, what are the concerns? Like, it sounds really cool and great. It has all these great health implications and and all these benefits, but it also has a lot of risk that people aren't talking about as much, and they need to. And here is, great. I won't show it to you, but this is fantastic. This is Jennifer Doudna. She is the co-founder of CRISPR technology. The reason why I have my job right now. And she goes through kind of what it is and how her and her partner, Emmanuela Charpentier, developed it. And she also has a great piece at the end, I think it's like 4 minutes, where she's like concerned, where she raises some of these concerns, and she calls them designer babies and says, the risk of this. And part of her concern is the reason why I'm here. So here is, actually, this is a CRISPR PSA project that I had my students do, and I'll show you what they did. So I did this as part of a unit. So I did this when students were learning about DNA, what DNA is, genes, chromosomes, all that, the central dogma, protein synthesis. So I put CRISPR right along there with all of that. So at the end of that unit, they had to create a PSA, a public service announcement.
Do you think teachers should integrate CRISPR into their curriculum?
Absolutely. Oh, yeah, 100%, because it is part of the teaching genetics. You have to talk about gene technology and genetic technology, and this is the wave, where there's been other genetic technology that have been similar to CRISPR, like TALENs, and TALENs is one. Another one is zinc finger. Yeah, ZFN. Yep, zinc finger, which the reason why CRISPR has taken off and why it's so talked about is because it's the cheapest and the quickest way. The other ones were a lot slower. They required a lot more money, and they only worked 3 base pairs at a time. Like zinc finger, I think only was able to edit only 3 base pairs at a time, which made it go very, very slowly in its editing process. Where CRISPR can work with, it doesn't, not just 3 base pairs, it can do hundreds at a time.
So it can do hundreds even though the guide RNA is only 20 nucleotides?
Well, so the guide RNA is, but so when I say hundreds, so it can so depending on that sequence that's being inserted, it will affect hundreds and hundreds. So the effect that it'll have will affect the genome, in the whole entire genome, which is billions of base pairs. But you can do a little longer than that. Even though the guide RNA, you can go a little longer and a few more base pairs. They're looking right now to see how many CRISPR edits you can make at once. So you can have modification, and it can work all at the same time, where it's multiple, but what does that look like? Does that increase room for error? And how does it increase room for error when it's red? Does it increase a mutation rate as well by doing that? So those are all the things that are kind of being experimented with right now, is putting multiple edits into a single genome at once. So that's why instead of just 3, yeah, you can do up to 20, but you can do it here, 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 and here, and can do them all at the same time. And what does that look like? And, what are the pros and cons of doing that? So those are all the kind of the things that are being done right now.
Who's your target audience at PGED?
So depends on who you're asking. So for me, my job is community outreach. So my target audience are members of the Boston community, mostly underrepresented groups. So the Latino community, the Black community, immigrant community. So people who normally would be out of school, might have been out of school for a long time, who don't have access to education the same way, where they might not be reading up on scientific articles or listening to NPR or, so making sure that I'm going to communities and to audience that otherwise might not hear about this stuff at all and not know it even exists. And if they wanted access to it, wouldn't know that it's even an option. But also our audience here at PGED is also schools and students and teachers. So we do professional development for teachers.
Like, what do you think the best means of communication is to try to educate the public about this?
So right now, I'm on the grounds trying to do workshops with people, where they are, community centers, even the faith-based community, getting to churches, talking to pastors, talking to imams in the Muslim community, and so kind of actually having conversations. That's one step. Another one is we do have a website, but people don't really know the website exists if you don't speak to them. No one's gonna be like, I want to Google PGA. They don't know what that is. Or I want to know CRISPR. You have to have heard about CRISPR to look it up. Yes. So I'm just trying. I think the first things first is word of mouth. Another thing is media. If we can get ourselves into the media, so I'm trying to work on getting into newspaper articles, good interviews. So trying to create or get an interview where our face is out there, or, whether it's the news, a public broadcast of some sort, so they can kind of see and hear us. And then that will hopefully drive interest to our website and drive interest in us doing further workshops.
So what are the goals at PGED?
So the goal is to inform, make people aware, and be cautious, and not immediately dismissive. Be cautious and aware, but not dismissive, just to not prevent further healthcare disparities. There's a lot of disparities between the haves and the have-nots, especially looking in communities of color, and people will see this and see the concerns, and there's a lot of historical concerns, and scientific ones from the past that people should be concerned with and should not forget, but I don't want that to be the driving force. People go, I will never look at, new technologies and science because of what's happened in the past. I want to know what's happened in the past, be aware, but also be understanding of what it's available and how it can help. And if you do want to use it, know what questions to ask. And how, and if you don't want to, that's your right too. That's fine too, but just be informed. So our goal is to inform, discuss, promote conversations, but to make sure people aren't automatically dismissive of the technologies, that if they do want them and do feel like it's something, when it is available in the near future, that they do have access to it because they know about it.